Good day, everyone. I am Eva May Villalahas, and I am here to explain the structure of the DNA and the step-by-step process of DNA replication. Before anything else, we should first define what is DNA. DNA, or deoxyribonucleic acid, is a molecule that contains the instructions an organism needs to develop, live, and reproduce. These instructions are found inside every cell and are passed down from parents to their children. The structure of the DNA. DNA is made up of molecules called nucleotides. Each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. The four types of nitrogen bases are adenine, thymine, guanine, and cytosine. The order of these bases is what determines DNA instructions or genetic code. Similar to the way the order of letters in the alphabet can be used to form a word, the order of nitrogen bases in a DNA sequence forms genes, which in the language of the cell tells cells how to make proteins. Another type of nucleic acid, ribonucleic acid or RNA, translates genetic information from DNA into proteins. Nucleotides are attached together to form two long strands that spiral to create a structure called a double helix. If you think of the double helix structure as a ladder, the phosphate and sugar molecules would be the sides, while the bases would be the rungs. The bases on one strand pair with the bases on another strand, adenine pairs with thymine, and guanine pairs with cytosine. DNA molecules are long. So long, in fact, that they can't fit into cells without the right packaging. To fit inside cells, DNA is coiled tightly to form structures we call chromosomes. Each chromosome contains a single DNA molecule. DNA replication in prokaryotes. Replication in prokaryotes starts from a sequence found on the chromosome called the origin of replication, the point at which the DNA opens up. Helicase opens up the DNA double helix, resulting in the formation of the replication fork. Single-strand binding proteins bind to the single-stranded DNA near the replication fork to keep the fork open. Primase synthesizes an RNA primer to initiate synthesis by DNA polymerase, which can add nucleotides only in the 5' to 3' direction. One strand is synthesized continuously in the direction of the replication fork. This is called the leading strand. The other strand is synthesized in a direction away from the replication fork. In short, stretches of DNA known as Okazaki fragments. This strand is known as the lagging strand. Once replication is completed, the RNA primers are replaced by DNA nucleotides, and the DNA is sealed with DNA ligase, which creates phosphodiester bonds between the 3' OH of one end and the 5' phosphate of the other strand. DNA replication in eukaryotes. Replication in eukaryotes starts at multiple origins of replication. The mechanism is quite similar to prokaryotes. A primer is required to initiate synthesis, which is then extended by DNA polymerase as it adds nucleotides one by one to the growing chain. The leading strand is synthesized continuously, whereas the lagging strand is synthesized in short stretches called Okazaki fragments. The RNA primers are replaced with DNA nucleotides. The DNA remains one continuous strand by linking the DNA fragments with DNA ligase. The ends of the chromosomes pose a problem as polymerase is unable to extend them without a primer. Telomerase, an enzyme with an inbuilt RNA template, extends the ends by copying the RNA template and extending one end of the chromosome. DNA polymerase can then extend the DNA using the primer. In this way, the ends of the chromosomes are protected. DNA is vital for all living beings, even plants. It is important for inheritance, coding for proteins, and the genetic instruction guide for life and its processes. DNA holds the instructions for an organism's or each cell's development, reproduction, and ultimately death. As what Lewis Thomas said, the capacity to blunder slightly is the real marvel of DNA. Without this special attribute, we would still be anaerobic bacteria and there would be no music.